Okay, moving on to objective number 2. List the subdivisions, or subdivisions, I guess, subdivisions in both microscopic and gross anatomy. So microscopic anatomy, those are ways of studying the structures of the human body that we can only see under a microscope. So we need something that can magnify with enough resolution greater than the naked eye can. Think microscopes mostly, and there's staining techniques and there's other things. Gross anatomy is basically saying you can see it with the naked eye. And that word gross, I don't know which one came first. I don't know if scientists or the early anatomists called it gross anatomy as in easily seen with the naked eye, or whether they started to think it was gross because dead bodies tend to smell. Anyway, I always kind of wonder when I see that where that term came from. The other way you could say this is, by the way, microscopic anatomy and macroscopic anatomy. In other words, big enough for you to be able to see. So let's see. Subdivisions of microscopic and gross anatomy. Let's start with microscopic since it's the first on there. And let's just say too small, or structures too small that are too, studying structures. Remember, we're in science, we're studying, so in fact, I'm going to put microscopic anatomy. Studying structures that are too small for the naked eye. And if I put some subdivisions in here, histology comes to mind first for me. And histology is the study of the microscopic anatomy of tissues. And tissues are collections of cells, by the way. To give you a strict definition of tissues, my favorite one is that a tissue is a homogeneous building material of the body. And there are 4 primary tissue types or major tissue types. Epithelial tissues, which make up linings and coverings. The outer surface of your skin is an epithelium, for example. The lining of your mouth is an epithelium, etc. Connective tissues, that includes a wide range of different tissues, from the only liquid tissue in the body, which is blood, all the way to bone tissue, which is the most brittle tissue in the body. Other connective tissues are cartilage, dense connective tissue, which comes in 2 varieties. Dense regular connective tissues makes up tendons and ligaments. And then dense irregular connective tissues, which we find in a lot of spots. Deeper in the skin, we find dense irregular connective tissue. The white of your eye is dense irregular connective tissue, and it's found in a bunch of other places. And then further connective tissues, adipose tissue is a connective tissue. So is areolar connective tissue, which is like adipose. It's another soft connective tissue kind of that fills spaces and that sort of thing. Anyway, that's connective tissues. So I got epithelial tissue, connective tissues, muscle tissues. There's 3 different types: cardiac muscle, smooth muscle, and skeletal muscle. All of these have one major thing in common, which is that they take the chemical energy stored in a molecule called ATP, adenosine triphosphate, and they can convert that chemical energy into the energy of motion through contraction of the muscle tissue. And there's a lot of other differences between smooth muscle, cardiac muscle, and skeletal muscle, but they all have that one thing in common. And of course, they're stimulatable. You can cause them to contract. And then we have nervous tissue as the last major tissue type. Nervous tissue is found in the brain, but in all other nerves and such in the body. spinal cord, peripheral nerves, etc. And nervous tissue is kind of like muscle tissue in that it is capable of changing its charge. If stimulated to do so, the cells in nervous tissue can change their charge and be stimulated. So because of that, they're called reactive tissues, or sensitive to stimuli, and end up changing their charge. Okay, sorry, I kind of went off track on what tissues are. So histology is the microscopic anatomy of tissues. Cytology is the study of structures inside of cells. Structures inside of cells. Includes things like organelles, like mitochondria, the nucleus, chromatin, which is the substance inside of the nucleus, which is DNA and its associated proteins, Golgi apparatus, lysosomes, peroxisomes, and other vesicular structures, or things that are made up of vesicles. But that's cytology. Those are the two major ones that I can think of off the top of my head. There may be more in your textbook, so you might write down more if you can. So that's microscopic anatomy. For gross anatomy, we study gross anatomy in various ways. The first one is just observational, without invading the body or anything like that. Observational surface anatomy, maybe. And then we can also do radiography and other imaging techniques. Uh-oh, I've misspelled something. There it is. MRIs, X-ray was the earliest form, and it's also the most risky. X-ray, CAT scan, which is X-ray at lower dosages of the radiation, and using computers to get a good image, a better image than X-ray. And then we got MRI, which uses magnet, basically a super powerful magnet, to cause the hydrogens in the water in your body to change their orientation and put out a signal that can be picked up by the machine, and then it builds a two-dimensional slice of the body. There's all kinds of imaging techniques that we can use to study anatomy. And then, of course, there's dissection. And I don't think at Kellogg we do any dissection, so we won't be dissecting anything, sorry, but we do have some dissections that are in the lab already. And the really neat thing about that is they're actual human specimens, which is really cool. Dissection, of course, is using a knife or scalpel to literally open up the body and to examine the parts. That's usually done on a dead person, of course, but some studies are done on living people if they sign up to be part of the study. If they have to have surgery anyway, then the surgeon can take pictures and study the parts that they come across along the way. Dissecting a living thing, by the way, is called vivisection, and we don't do that to humans ever. Well, not in the U.S. anyway. Historically, it's been done. During World War II, it was done on some hideous occasions. Anyway, I think I've covered this one in pretty good detail, so I'm going to move on to the next one.